Hello, welcome to Chapter 8. This is part of our fourth unit here. Chapter 8 and Chapter 9 go well together because we're going to talk about my favorite subject, which is genetics, DNA, biotechnology, these things. These are the things that I really love in biology and I think are cool. We are going to talk not only about bacterial genetics, but how we use them in our everyday life. You might not think it, but there are a lot of things in our everyday life that are affected by biotechnology. Particularly food is a form of biotechnology, if you will. But we'll talk about some very specific techniques, and we will actually do some of these things in the lab, so it's pretty exciting. The main problem that I find when talking about working with DNA and proteins and things like this is we have a hard time picturing the scale that we're talking about. We're talking about tiny molecules that you can't see under a microscope, right? You have to maybe use an electron microscope to even see these things, but even then, they're really too small to see. So how do you go about working with something or studying something that is so small? It would be very handy, and we would be able to do a lot, right? If we had little tweezers that we could use to cut and paste pieces of DNA. But that's not the case, right? To do this, people have had to be very clever, and they've come up with clever methods to steal things from bacteria that we can use to cut DNA and then move it and then paste it into places. Sequencing DNA, copying DNA. All of these things rely on tools that are taken from bacteria. So bacteria were really the model for studying DNA and proteins and things like this. And then we stole all of those ideas really from nature and we use them in our lives for biotechnology. In this chapter, we're going to talk about the structure of bacterial genomes and how they get replicated. We'll also talk about this related topic called plasmids, which are of great importance to healthcare. We'll talk about biotechnology processes that kind of help with medical diagnoses, but even beyond that, really just our general basic research that we need to do to learn about things so that we can find applications for these things. So we'll talk about restriction enzymes, gel electrophoresis, what's called recombinant DNA, and of course we'll talk about PCR, the polymerase chain reaction. We will actually get to do this in the lab, which is super cool. We need to talk also about the processes of transcription, which is taking DNA and turning it into RNA, and then translation, which is taking that RNA and turning it into a protein. This is related to a topic called gene expression, which means to make the proteins that are coded for in a gene. And gene expression doesn't just happen randomly. It's tightly regulated. Bacteria and you need to turn certain genes on and certain genes off depending on the scenario, right? The environment, what you're doing, what you've been eating, things like this. So these are the critical points that we'll be covering in this chapter. Let's start with a case study here, and this is really about using DNA technologies to diagnose a problem. So we have Tina, she's 33, and she's having her first dental exam in 5 years. That's a bit of a long time to go, but it's understandable, right? People hate going to the dentist, life is busy, it can be expensive. So she goes in and she tells the hygienist that her gums hurt and that she sees stains on her pillow where her jaw is resting. Of course, the hygienist asks about a general health background. Tina says that she doesn't smoke and doesn't drink. Now, this is a bit of a problem here. The hygienist looks at her mouth and we see major issues here. So we see swollen gums, they bleed when they're probed, and you've probably noticed when you go to the dentist, they do that little thing where they talk about, they go 3, 2, 2, 3. They're measuring the depth of your gums there. And in Tina's case, she has at least 5 millimeter deep gum pockets in there. So an x-ray actually reveals that she's starting to lose bone. So the dentist tells her that this is called periodontitis, which means inflammation of the gums and the bone surrounding the teeth. And that inflammation, in this case, is caused by a large amount of dental plaque. So all this yellow and white material that's in here, that is dental plaque. And as we talked about in the last section, dental plaque is a biofilm. That biofilm has that sticky matrix, and inside of it are bacterial species. In this case, it's a mixture of bacterial species. So without regular brushing and proper oral hygiene, this biofilm grows and builds up, and it secretes acids and other things that start to decay the teeth and can cause loss of teeth ultimately. Now, at this point, the patient finally admits that yes, they smoke a pack a day. So it's not just that they don't take care of their mouth, they're doing an activity that is poor for their oral health. So here's a great point about working with human patients. Humans can lie, which is difficult when you're doing either scientific research or trying to diagnose something or just get a straight answer from someone. People get embarrassed, they lie, and that makes medicine all the harder. So be honest with your healthcare professionals is the takeaway from this. They generally know, right? You can tell from this. So lying doesn't really help you in any way. So to treat this, we have a problem. We want to identify what microbes are in there. And biofilms are a mixture of microbes. You could take a sample, grow it, streak it out, do all these tests. But we actually have quicker ways of identifying microbes. One of which is a PCR test. Now, you've probably heard of PCR quite a lot now because of COVID testing. We'll talk about what PCR is and how it works, but basically it's a way of copying DNA. And we can copy specific pieces of DNA, which allows us to see, is a microbe there or is it not there? So PCR tests, we can copy pieces of DNA, and then we can even send those pieces of DNA to be sequenced. So if we sequence pieces of DNA, we get the full ATGC content of it and the order of it, and actually we can put that into a database and identify specific strains of bacteria using that. So PCR and DNA sequencing allow us to find that in the plaque there's two bacteria. I'm gonna butcher these. These are tough ones. Porphyromonas gingivalis, which probably causes gingivitis, another mouth disease. Aggregatibacter actinomycetum comitans. I promise I didn't do that 5 or 6 times trying to pronounce these correctly. This is a terrible name. These two microbes, they're gram-negatives, they're anaerobes, so they're living in an anaerobic biofilm inside of there. And because of the nature of them, we know that they're sensitive to these two antibiotics, so the dentist will prescribe them to try and clear up the biofilm. Biofilms are really sticky and hard to deal with, so just brushing probably won't deal with this. They're probably also starting to get into the bloodstream, so there's infection of the tissue in there, so antibiotics are definitely warranted in this case. So that's a use of DNA technology, right? Sequencing those species that are in there allows us to identify it. And we've talked about finding and identifying bacteria before, but that all required growth. And biofilms are really hard to grow and deal with. So this DNA sequencing is quick. You can get it done in an afternoon, really, if you have the time. So we need to talk about some important definitions that are going to be key throughout this chapter and the next section as well. We need to talk about this term, the genome. You've probably heard the term genome, but it has a very specific definition. The genome is the unique set of all genetic material, the DNA, in a cell. So in your cells, you have your chromosomes that are all there. You have 23 different types of chromosomes, but you have 2 copies of each, one from the mother, one from the father. Those end up making up your genome, right? All that genetic information in there is your genome. And each cell has its own genome. Every cell has a copy of the genome in it. If we compare organisms in a species, their genome will be very, very similar. So your genome, your siblings' genomes, your family's genomes, and really you and every single person on Earth, you only have tiny, minor differences. You might think that people are super diverse and different, and they are on the surface, but at their genome level, we're all super similar. And there's minor differences between there. But those minor differences can mean a lot of big differences. So when we talk about differences between bacterial strains, things like one E. coli strain that makes you sick and another E. coli strain that lives in your gut naturally as part of your microbiome, the differences there are very, very small, but very, very major in terms of what it means for our health. So the genome is all that DNA material. A gene is a region of DNA that contains information that codes for a protein. So in the DNA, there are regions that have information that if you decode them, it codes for a protein. And we will learn how to do this. So you're going to learn how to read DNA in this course, which is pretty awesome, honestly. Like, that's decoding life right there. Because of the kind of uniqueness of each different organism, we can find ways to specifically identify microbes using like DNA sequencing and other molecular techniques. So we can look at the genes, we can look at the genome and say, yes, this is this organism specifically. And we can even say this is a specific substrain of it. So you're probably familiar, we can use DNA technologies to identify a specific person as well. So these are 2 critical terms to have down because we're going to use them quite a lot in this chapter. We also need to talk about something that is called the central dogma of molecular biology. So we're talking a lot about molecular biology. I consider myself a molecular biologist. That's what I did mainly when I was a researcher. I studied DNA and proteins. Those are molecules of biology, so we call that molecular biology, related to genetics and things like that. This central dogma is that there's a flow of information that happens. We have information stored in the DNA that's going to get converted into RNA by a process called transcription. And that RNA is an intermediate that's going to go out and go find a ribosome, which is going to convert that information into a protein through a process called translation. Okay, these are easy to confuse because they both have trans in them. Transcription is DNA to RNA, and translation is RNA to protein. We'll talk about each of those processes in detail. So what's in a gene? Genes code for proteins. Many of these are enzymes, and you share common genes with lots of different species. In fact, you have common genes with bacteria. Think about the enzymes that go through that glycolysis process that we talked about in the terrible metabolism chapter, right? You do glycolysis almost identically to a bacteria. And that's because we have a shared evolutionary lineage, right? We all had a common ancestor at one point, and we've evolved and developed different genes, but we still have some of the same genes. The sequences are slightly different, but we still have common genes that do the same thing. Many pathogens often have very unique genes that code for what we call virulence factors. These are things that allow them to infect and attach to the host and maybe cause the signs of disease. We'll talk about some specific ones in this chapter as well. Microbes that live in the environment. Think the extremophiles and things like that, things that live in boiling sulfur pools or things like that, they're going to have unique genes that enable them to live in those specific climates or conditions. Halophiles, which live in salty environments. They're going to have specific pumps that code for proteins that pump salt out of their cell, right, so they can survive in the salty environment. When we have changes in the genetic sequence of a species, we call this evolution. There's a whole field called population genetics that studies this, but really as genetic sequences change in a population, that is the process of evolution right there. And we're going to see evolution in action in microbes. There are very key examples of where evolution is happening. So the goal of this chapter is, first we need to learn about the composition of a genome, and then we're going to learn about how the genes in the genome are expressed or turned into proteins. We're then going to take that knowledge and look at how we can exploit bacterial processes and viral processes to create tools. We're going to steal the pieces that naturally go on in this and use that to help us detect and diagnose illnesses, and then ultimately develop therapies that can help with these things. So 8.1 is all about the genome. We need to talk about the structure of it and explain a bit about how it's different from eukaryotes. So your genome is composed slightly differently and organized slightly different than a bacteria. Then we're going to talk about this small pieces of DNA that we call plasmids. And plasmids have really key roles in pathogenesis, the ability of microbes to be pathogenic, but also the ongoing spread of antibiotic resistance, which is a huge problem in healthcare right now. In a bacteria, there is a genome that is composed generally of a single but very long circular chromosome. So it's a big circle of DNA. And this is generally several million bases. So there's several million A, T, G, and C's long. So I'm going to use this term bases. That is an A or a T or a G or a C. And we'll see how they're arranged in there. But in this case, right, this is an image of a bacterial cell, and it's been broken apart, and all of its DNA is spilling out. This whole messy tangle here is one big circle of DNA that's a million bases long, and it's been stained, so it's kind of able to be seen here. So in that genome, there are regions called genes. Those genes code for proteins. Not all of the DNA codes for proteins, but in bacteria, most of the DNA codes for proteins, and they're going to have a couple thousand genes in there. The chromosome itself is a double strand, so the double helix of deoxyribonucleic acid, or DNA. And we're going to learn about the structure of DNA right now because the structure is intimately linked with how DNA copies itself and how it is transcribed. So we'll also do some lab exercises that will reinforce this. Okay, don't freak out. This is much simpler than it looks, I promise. And again, it's just chemistry light here. So this is a molecule of DNA, and it would continue going. This is a very short molecule of DNA if it's only 4 bases long here. Deoxyribonucleic acid, right? Remember back to our biomolecules, we have what's called a DNTP. This is a phosphate attached to sugar, deoxyribose in the case of DNA, and then it has what's called a nucleobase on it. This is where we get the A, T, G, or C part. That is the information part there. So DNA is a double strand of these molecules. So they run in opposite directions, as we'll see. This one goes this way from what we call the 5' end to the 3' end, and the other side goes in reverse. It's what we call anti-parallel. And ultimately, the structure forms this twisted kind of ladder shape, or a double helix, we call it. Remember, Rosalind Franklin's image of that X from the X-ray crystallography indicated that it was helical in shape. And you'll notice that on the outside are the sugar and the phosphate, and on the inside of the double helix are the nucleobases, the nucleotides in here, the information piece. So each one of these is a nucleotide or a DNTP, and the information is stored in these letters here. So A, and then this is a G here, and there's a C over here. In DNA, A bases always pair with T on the other side, and G always pairs with C. So that's really convenient because if you know one side, you automatically know what the other side will be. Seems very inefficient, right? Why have this duplicate information? Well, think about this, right? You've probably been there. You have, it's late at night. You probably didn't wait until the last minute to do your assignment, but let's just say you did. I know you wouldn't do that, but you waited till the last minute to do your assignment, and your computer crashes. Did you save a backup copy of your assignment? If you didn't, you're in big trouble, right? But if you did save a backup copy of your assignment, you don't lose all your work, right? That's what DNA does. DNA has duplicates, right? So it's got the information on one side and a backup copy on the other side. So that's really critical. That's why DNA is our genetic storage material. In between here, these A's and T's and G's and C's, they're held together by hydrogen bonds. We'll talk about why that matters in a moment. So if we zoom out, this is kind of a common graphic for representing DNA. This is a cartoon of that double helix. And now we're going to talk about what information is contained within there. In the genome of a species, bacteria have genes in their genome, right? The genome is all the DNA, but it's organized slightly different than in a eukaryotic organism. In bacteria, things tend to be compact and very efficient, right? Bacteria are all about efficient replication, going fast, live fast, divide fast, right? So bacteria have very dense genomes that have lots of these coding regions. So this is the information that will make a protein. They have a lot less what's called non-coding DNA, which is the gray parts here. In your genome, you might have a little bit here, some non-coding in here, a little bit here, non-coding, a little bit here. These non-coding bits in a gene in your genome are called introns, and the coding parts are called exons. A lot of times, for a long time, people thought that this non-coding stuff was junk DNA. It didn't have any purpose. But we now know that it's really important in regulating when this gene gets expressed or gets made into a protein. This non-coding DNA has functions in that. So in the bacterial genome, you see that these what are called introns are missing. It's just that dense coding information just right there. So that's the big difference between eukaryotes and prokaryotes in terms of their genomes there. The process of gene expression is turning this gene into a protein. That's ultimately what gene expression is. Most genes have a region in front of them that doesn't actually code for protein, but it's called a promoter. That's the little red bit here. This promoter is really important in helping determine when the gene gets turned on. For simplicity's sake, we're going to assume that one gene codes for one protein. This isn't quite true. This is the little white lie that I'm going to tell you. But for our purposes, that's what we're going to go with. So this promoter helps regulate when the gene is going to be expressed. And gene expression has a couple of different steps. We have transcription, which takes the information in the DNA and copies it, but turns it into a different form called RNA, particularly mRNA. Then we have translation, which is turning the mRNA into a chain of amino acids. And then remember, those amino acids, they need to fold into our protein. So this is the process of gene expression. This is going to happen at different times under different scenarios. A bacteria moves to a new environment and there is the sugar lactose there, right? The bacteria is only going to make the genes to break down lactose when lactose is present. So that's an example of gene expression right there. In addition to the main chromosome, bacteria can have other little pieces of DNA called plasmids. So plasmids are little circular pieces of DNA that are much shorter. There is a typo in this image from our textbook. This says that the main chromosome is 5 million base pairs long. So it's 5 million A, T, G, and C's long. Okay, that makes sense. But then it says these plasmids are 200 kilobases long, which is incorrect. It says 200,000 kilobases. It should be 200,000 base pairs. So I apologize for that error. Probably doesn't affect your life, but it always bothers me. I need to fix this figure. These plasmids, this one is 200,000 bases compared with 1 million here. So much shorter. But what's on these plasmids? Well, plasmids are circular. They can replicate separately from the genome. And the critical part is that plasmids can get shared from one bacteria to another. On plasmids, we tend to find genes that are important for things like living in a specific environment, or antibiotic resistance, or making something a pathogen. Some bacteria can transfer plasmids that turn a non-pathogenic bacteria into a pathogenic bacteria, or turn a antibiotic susceptible bacteria into one that is now resistant to an antibiotic. That's why we're so concerned about these plasmids, because they're so easily shared between individuals. That's like me giving you some of my genes, and now you can make different proteins. So the bacterial DNA in the chromosome is there, and then there's a bunch of plasmids in here. Now, plasmids are interesting from that kind of natural perspective, but plasmids are also very useful to us. So plasmids often contain genes that help organisms live in a specific environment, and you can share them between organisms. Sometimes they have antibiotic resistance genes. Sometimes, though, we have found clever ways to take a plasmid and we can paste DNA that we're interested in studying into that plasmid. So one example that I've actually already shown you in lab is GFP bacteria. So I've taken a plasmid and I've put a protein that makes a green fluorescent protein on it. And under a UV light, if I put that plasmid in, the bacteria make this fluorescent protein, and under UV, they glow green. So we'll actually look at this again in the lab. But plasmids are really useful for a molecular biologist because we can use them as a tool to play with DNA and proteins. If you're concerned about healthcare, they are often very difficult to deal with because when a bacteria bursts, you maybe kill it with something like a cleaner. It leaves behind those plasmids, and if other bacteria come along, they can actually suck them up and acquire those new genes. So let's look at an example of how we might use this information. So we have Marianne. She's recovering from a kidney stone operation, and she has a tube that goes through her body into her kidney to help drain her kidney. So if you know anything about medicine, anytime you puncture the human body, there is a risk for microbial infection, right? That's a huge risk. So having this hole in you is definitely something that needs to be watched, right? We watch for signs of infection here. Marianne develops what's called endocarditis, which is a heart infection. And so she's transferred to the hospital where 5 days later, she is experiencing what's called septic shock, which is where there's bacteria growing in the blood, and it causes various symptoms that are related to shock. She ultimately has to be intubated to help her breathe. So we need to figure out what the pathogen is here. And let's think about what the first thing we should do to identify this is. Well, the first thing we're going to do is we need to make some cultures, right? We're going to take blood and try to figure out if there's bacteria in her blood. We're also going to take other fluids like urine and test that. And in this case, we find that her urine contains this bacterial pathogen called Klebsiella pneumoniae. This probably came from the kidney tube. And to identify this, we probably use that PCR and that DNA sequencing to figure out exactly what it is. Now, here's the problem. Klebsiella pneumonia is a very common hospitally acquired pathogen, and it has become often very drug resistant or extremely extensively drug resistant, XDR. So we're going to test with different antibiotics on our culture, and we find that in this case, yes, she does have a drug resistant strain. We test 6 different antibiotics. We find only one of them works even mildly. That's not good news. So because we have no way to kill those microbes, after 15 days, this patient is unable to clear the infection. Her immune system cannot deal with it, and she ends up dying from this. So Klebsiella pneumonia is one of 6 different pathogens which are the leading causes of hospital-acquired, life-threatening infections. We call them the escape pathogens. These are Enterococcus fascium, Staphylococcus aureus, which you've probably heard of with MRSA, Klebsiella pneumonia, the one in this one, Acinetobacter baumannii, Pseudomonas aeruginosa, we've talked about that in cystic fibrosis patients, and other Enterobacter species in there. So these are commonly found in hospitals, and they often share plasmids with one another. And on those plasmids can be genes that cause resistance to antibiotics. So in Marianne's case, unfortunately, she acquired a strain of Klebsiella pneumoniae that had resistance to 5 different antibiotics and was only mildly susceptible to one of the 6 that were tried. So these plasmids get passed between bacteria. And this is one of the reasons that keeping hospitals clean and knowing proper decontamination and sterilization techniques is so critical, because even just killing the microbes isn't enough. We need to remove the DNA from there so they don't get transferred between individuals. These plasmids are so easily transferred that one day a bacteria that is not resistant to antibiotics can quickly become resistant through transferring of these plasmids. I promise it's not all doom and gloom in this chapter. Yes, it's very important, but there are fascinating things in here. So we talked about the genome. This is the total DNA content of a cell. Bacterial genomes are the main circular chromosome and any shorter DNA molecules on plasmids that are there. Chromosomes of bacterial genomes, they're typically circular, whereas yours are linear. Plasmids are small circular pieces of DNA that contain supplementary genes. They might encode a gene pathway that allows the bacteria to degrade materials from their environment, or give them antibiotic resistance, which gives them a fitness benefit, as we call it. These plasmids, scarily, are transferable from one bacteria to another, and even from one species to another. And they can transfer those genes, and that allows pathogens to rapidly evolve different capabilities, like resistance to multiple antibiotics. So this is a very scary proposition if you're a healthcare provider. And then we have common pathogens that are considered very dangerous because they're extremely drug resistant, and they're often acquired in hospitals or healthcare facilities. Plasmids play a role in the evolution of these pathogens, so understanding them is very critical. All right, that's it for 8.1.